Thank you.
Welcome, everybody, and good morning. Welcome to the Informatics Institute PowerTalk Informatics seminar series. Happy Friday, and also happy Mardi Gras, or happy beginning of Lent, whatever you celebrate. We're very privileged to have today's speaker, Dr. Zheng, professor, founding director of the Data Science Informatics Core for Cancer Research, and the director of the Bioinformatics and High Performance Computing Service Center, which is a CTSA-funded CCTS, and associate director for the Center for Computational Biomedicine at the UT Health Houston School of Biomedical Informatics. He completed his MS in computer science from UT Dallas and Ph.D. from UT Southwestern. He spent most of his career in bioinformatics research in both industrial and academic settings, and which pretty much touches on all the major parts of informatics, his career, and his research interests are integrating, modeling, visualizing, and mining eukaryotic genome information with translational medicine, and pursuing novel approaches for knowledge representation. He's developed frameworks to integrate and visualize eukaryotic genomes in 3 dimensions, and the concept of ontology fingerprints to embed ontology term embedding for distributed representation of genes, computational for mining the biomedical literature, so that helps to make sense of a lot of information. Also deep learning approaches to predict effective drug combinations, and for biomedical research. His current research focus developing novel data mining methods to extract information from biomedical literature for their therapeutic strategies against cancer, Alzheimer's, and other diseases. He's on the editorial board of 2 bioinformatics journals, and his research is supported by multiple funded programs. And I'm very happy to introduce him. His title of his talk, Distributed Representation of Biological Concepts.
Great. Thank you very much, Amy. And I really appreciate Jake sort of giving me the opportunity to present to you guys. Today, I'm just going to talk about these particular projects I have been working on for many years, focusing on distributed representation of biological concepts. I'll start with, as Amy mentioned, that on these ontology fingerprints, which is a distributed representation of genes and the diseases using the ontology terms. Yeah, then I will talk a little bit about these applications of this approach, and then I'll talk about the latest advancement in that, taking the advantage of learned deep representation, and I also touch a little bit on the recent competition we participated, as well as this data science and the informatics core for cancer research. Let me start with this explicit distributed representation. So here, explicit also modifies representation, not the distributed. So you'll see why. If we think about biological research, we help us to understand what a gene does. At the beginning, we have the concept of one gene, one enzyme, one function. So you label the gene with the annotation. In this case, this gene APOC2 is apolipoprotein C2. From the name, you can guess what the protein is about. But then as we expand our research, we know more about genes, and then we start to describe the genes in more detail, such as this summary at the NCBI gene page. And then people realize that this kind of text description is not computationally friendly, and people start to develop more rigorous terminologies like gene ontology to describe the gene functions, process, and the components. And in addition to that, then we also know more and more about the other aspect of the genes. So you have things like gene card that have a comprehensive information about a particular gene, including variants, mutations, their genomic location, gene expression, et cetera, besides the annotation of what the gene does. But these kind of annotations, while it can help us know the function of the genes, there are two challenges. The first one is the volume challenge. As you can see, the literature that published over the years is growing almost like exponential. So there's so many papers published on every aspect of the genes, drugs, disease, and other aspects of biological system. It's very difficult to keep up with, the literature. And right now, the high quality, reliable annotation about these biological concepts is still mainly a manual process. Computational approaches start to play more and more important roles, but the quality is still not comparable to the human manual annotation. So right now, the high quality annotations are still limited to the publications read by the curators, and also the annotation for a gene is still limited, still has very limited amount of information. And to just add to this challenge to impress you, here's me as a graduate student. That's like a paper I read every semester. It's getting crazy. So the second challenge is the computability challenge. There is no quantitative measure to assess this kind of annotation we have currently. For example, here you have gene ontology function annotation for 3 genes. You have ApoE, you have APO2, and you have TP53. You can see that each gene is described by a set of ontology terms, but among all these ontology terms, which one is the major function of this gene? Which one is more like associated function of this gene? And how important are these functions that might relate to each other? So there's no such measure to tell you these kind of detail, which is very important for us to perform any computational analysis. The other challenge about the computability is there's no measure to infer gene-gene relationship. You can see that, the top 2, ApoE and APO2, there are, certain amount of overlap in terms of gene ontology term used, but there's no measure to sort of help you to calculate to what a degree the similarity is, and how that reflect how these genes might be related or have performed similar function. Also, for the TP53, obviously you can see that the overlap is much less compared to the top 2. But again, it is difficult to assess whether the gene at the bottom is related to the top 2 genes or not. So this kind of challenge in terms of computability really limited our ability to infer novel gene-gene relationships. Currently, there are certain computational ways to use this kind of annotation. For example, gene set enrichment analysis for gene expression, transcription profile, as well as proteomics. But in terms of inferring directly how genes are related based on what we know about their function, is still limited. So because of that, we came up with this idea called ontology fingerprints as a solution for these challenges. The basic idea is that we take a text mining approach to identify ontology terms enriched in the PubMed abstracts that are relevant to a particular gene or disease, and then calculate their enrichment p-value as a way to represent each gene. So here is an idea. Here is how we did this. So we took PubMed abstracts and identify PubMed abstracts that link to a particular gene. So we can count how many abstracts relevant to a particular gene. We can also identify gene ontology terms in these abstracts. And then we can create this 2x2 table where we have the number of abstracts related to a gene and also contain that particular ontology term. And then we also know the abstracts that's not related to a gene, but also have that, but still have that ontology term. And also we have abstracts without that term, but relevant to a gene, or we have abstracts that has no mentioning of that ontology term, and also is irrelevant to this particular gene we're interested. So based on these 2x2 table, we can calculate the enrichment p-value to see whether any particular ontology term is enriched in the abstracts related to this gene. And we do that for all the gene ontology terms, and we have a list of the p-value that for each of these ontology terms. So this is what we call the ontology fingerprints. This is done way before deep learning or word2vec, those kind of things, which I will mention later on. So with all that, basically we got what's called the distributed representation. There are three defining characteristics of distributed representation. The first one is it's distributed, right? So if you look at this representation, basically it is a vector, a numerical vector with all these enrichment p-value, right? And each dimension of this vector has a solid value. So that's important because you are representing this concept, this gene, by high dimensional vectors instead of just a single aspect. For example, oh, it's a lipase. It has a lipase activity. So that way you have a more comprehensive representation of what the gene actually is. The second characteristics is the distribution. This representation is based on distributional statistics. So basically, if you look at each dimension of this vector, it's an enrichment p-value, which considers the distribution of this particular ontology term in all the literature, based on the consideration of the gene and the number of PubMed abstracts that relevant to this gene and relevant to this ontology term. So this kind of distribution is also important because it's really a very important characteristics of this gene. And the third characteristics of this distributed representation is it's embedded in a low dimension vector. So imagine that if you truly want to represent a gene, right, using the literature, you would imagine that you will calculate this enrichment p-value for all the words in the literature. That is a fully comprehensive representation of that gene. However, as we know, many of these terms are not really biologically important, or it's not really relevant to the gene function or biological process or cellular component. So instead of have a long vectors of all the words appeared in biomedical literature, we decide to focus on the gene ontology. And so that the vector that represents this gene has a much smaller dimension. And so this is a dimension reduction achieved by domain ontology. And these three defining characteristics is also important for the later on deep learning based approach, or the new approach such as GLAB. So these are the three defining characteristics that's shared by all these different distributed representation. Here I just want to emphasize why we call it distributed representation. It's sort of relevant, it's sort of in contrast to one-hot encoding. So here I give you an example of four-word phrase, right? SMARCB1 deficient bladder cancer. And here you can see that each of these words is represented by a vector. However, in this what's called a one-hot encoding, each vector only have one value. That is, here, if it's SMARCB1, the vector only has one that corresponding to SMARCB1. All the other dimensions is 0. Likewise, deficient, it has 0 in all the dimensions except 1 that corresponding to the word deficient. So this kind of representation is called a one-hot encoding. And obviously you can see that if you want to use one-hot encoding to infer relationship by compare this representation for two words, you cannot do that because all the other dimensions are zeros. So there's no way you can infer how these two are similar or dissimilar. On the other hand, if you look at the distributed representation, each word is represented by a vector of many different values in different dimensions. So by doing that, you actually can compare the vectors representing two different words and see how they are similar to each other. Based on that similarity, and then you can infer whether these two words is related or not. So that's how, what's behind this whole idea of distributed representation for, biological concepts. And that's what we do. We did for the ontology fingerprints for genes and the disease and the drugs. So once you have, as I mentioned, once you have these distributed representation vectors, then you can compare these vectors to calculate the similarity by looking at where, how many dimensions they match, and also the value enrichment, the p-value, and calculate something, in this case, a modified version of the dot product for the similarity score. So the idea is that if two genes has very similar vector representation or ontology fingerprints, then it's likely their function are closely related. On the other hand, if two genes have very dissimilar ontology fingerprints, then it's unlikely they're biologically relevant. Imagine that, for example, if two genes belong to the same pathway, say cell cycle. So you can see that if people publish papers for these two genes, it's very likely they will mention many ontology terms that is related to cell cycle. So that when you calculate the ontology fingerprints for these two genes, it's very likely these two genes are populated with a significant enrichment p-value for those ontology terms for the cell cycle. And if you compare these two fingerprints, you get a very high similarity. That's the basic idea about these. Once you do this pairwise comparison, then you can generate adjacency matrix. So here you can see you have, for example, 25,000 genes in the human genome, and you make this table where all the columns are genes, where the rows are also genes, and then you calculate the similarity score between genes, and then you generate this table. So for those who are familiar with transformer model, you might see this familiar. So this is basically a self-attention table, right? So you have an adjacency matrix, and then you can generate a gene network. So in this gene network, you have dots, which are genes, and then you have the edges that corresponding to these similarity scores. So genes who have a higher similarity score will be close to each other, where genes who have a very low similarity score, they will be further apart. So you can imagine that based on these, the genes belong to the same pathway, they actually cluster together in this network, where genes that have nothing to do with each other, then they will be further apart. And so you can see this subnetwork structure that corresponds to the biological function. And also there's a few pathways, for example, like the ubiquitin pathway that really involves that's relevant to many different biological processes. So you will see that genes that belong to these kind of pathways, they sort of like scattered around the whole network. So that's how we basically take distributed representation of genes using ontology terms, and then compare these distributed representation between genes to generate this network. And once we generate this network, then we can do many things. Here's one example of what we did. So imagine that in this network, you find out, okay, all my genes belong to this particular pathway. It's called sphingolipid pathway that cluster together, right? However, if you look closer, you will see that besides the known genes that belong to this pathway, you also see some other genes that also belong to this subnetwork structure. So what are those genes? Are they really functionally relevant to this pathway? So here's what we did. We took these clusters that belong to this sphingolipid pathway, and then we look at the known sphingolipid genes, and because it's well-studied pathway, we know how these genes are related to each other. They encode different proteins that catalyze the biochemistry reactions for this particular pathway. So we took this pathway, and then there's a way to monitor the pathway activity by treat the cells. Oh, by the way, this is in yeast as a model organism. So we took these drugs called myriocin, which will block a particular enzyme of this pathway. And so the in vivo activity of this pathway can be monitored by the sensitivity of cells to this particular drug. And so now we have a system to monitor the pathway activity in vivo. Then we start to test what are the other genes that's not known to belong to this pathway, but they sort of clustered into this subnetwork structure that belong to this pathway. So we knocked out some of these genes, and then we measured the sensitivity of these strains without these genes and see whether the in vivo activity of this pathway is changed. And here you can see that here's the wild type that has a certain degree of sensitivity to this drug. And then you have a negative control where it's very sensitive to this drug. And then here are the two genes that nobody have any idea they belong to this pathway. However, if you knock them down, you can see that cells become very sensitive to this drug, indicating that if you knock down these genes, if you delete these genes, the in vivo activity of this pathway has been dramatically changed. So this is the indication that these genes actually will have impact on this sphingolipid pathway in vivo. And we did this for the top ranked genes that cluster together with the sphingolipid pathway genes, but there's no prior knowledge that they have anything to do with this pathway. And overall, we identified and validated 14 of these novel pathway players. That's how we call it NPP, novel pathway players. And out of these 14 genes, what's interesting is that if you take these 14 genes, and then you take other algorithms that predict the new pathway genes or players using different data sources or different approaches, and very few of the genes we identified can be predicted by these algorithms. So you can see that, this approach is quite different or orthogonal to the existing methods that people developed. So the indication of these, there are two indications of this. One is that this approach can predict novel pathway players, other methods cannot play, right? But what's also interesting, if you think about this, is that you have a known pathway, and that pathway is targeted by a known drug, which, at a certain concentration will kill cells. And then you use this network, you identify other genes that if you knock down this gene together with this drug, it will kill cells more efficiently than if the drug acts alone on the cell. So that means this network can be really a good way to identify components for drug combination therapy, right? So you identify the new target, like in this case, one of these genes. And if you have another drug target this gene, the same effect as you knock out this gene. And in combination with your existing drug Mariocin, then you can kill cells much more efficiently. So we look at this, we say, well, this might be a good way to identify a combination therapy for cancer treatment, right? So we have a known drug target pathway, and then we can use this method to identify new targets. And if this target has an existing drug, then we use both drugs that might kill cancer cell more efficiently. So based on this idea, then we developed a drug combination prediction algorithm based on deep learning architecture. It's called stacked restricted Boltzmann machine network. So the idea is that we took the cell line information, which is transcription profiles of individual cell lines. And then we took our ontology fingerprints. And then we took two drugs that target pathway genes with qualified first label. And then we feed this information into this deep neural network. And then we predict whether these two drugs have a significant synergy score as a way to identify efficient drug combination for cancer therapy. And sure enough, the performance at that time is much better than state of the art. But certainly now they involve much more. We'll talk about that later. So, but overall, you take this different information, you integrate them through a deep learning network, and you can have a good performance to predict drug synergy with this distributed representation information. So here, the data we use is from AstraZeneca, the Dream for Competition, where you have almost 5,000 pairs of known drug combinations. 3,200 has a synergistic effect, 1,800 are negative, and we can detect, 1927 pairs as synergistic and 1030 pairs as negative. So that's how we evaluated our performance. So this distributed representation for genes, we use the ontology terms and we use literature, but we definitely not just limit ourselves to that. We actually expand that. We can use other ways to develop distributed representation. In this case, we develop what's called a gene fingerprint model. So instead of using ontology terms, now we use genes appeared in the literature to represent the disease. And by the same approach, we calculate the enrichment p-value of genes for each disease, and then we generate a primary matrix. After we generate primary matrix, then we took some matrix operation to optimize the representation, generate this distance matrix, and then based on this distance matrix, we performed spectral clustering to identify new disease-to-disease relationship. So for the matrix optimization, we use this we use a single value decomposition and also use a low rank approximation, and to generate this distance matrix of disease based on the Spearman correlation, and then we perform the spectral clustering. And here we also looked into the number of genes that are related to disease and how they perform. And here's the F1 score. By using this approach, we inferred what are the other diseases that might be related to COPD. And here are several diseases that we identified that's related to the COPD that in literature, there's no such information. And so after we identify these, we actually have the physicians to evaluate these, and they gain some novel insight why this disease could be relevant to the COPD. We also applied the same approach for cancer and look into the pathways and genes and the treatments, and identified many important genes that could be targets for cancer. And so far, I am only talking about this explicit embedding for distributed representation, right? So this distributed representation, it is based on the distributional statistics manually calculated. Yes, in this case, we use enrichment p-value, and it's not learned. So it's not like using neural network to learn from the data what is the distributional statistics for each dimension. The other thing is that what we did, the distributional statistics at the document level. As you may remember, what I mentioned is that we look at the co-occurrence of ontology terms and the genes in the same PubMed abstract. So we only concern about whether two concepts appear in the same document. And we're not looking at the sentence level, which is more important in terms of the context, the biological details, and the semantics. Another limitation is we use ontology terms. Obviously, this is developed by human being. It's high quality, but it's not a full or comprehensive representation or captured by all aspects of biology. And the other challenge is that ontology terms are developed to precisely describe biological process, cell gene functions, and also the cellular components. They are not really sort of geared toward sort of like a usability or readability. By that, I mean, when people write papers, they don't sort of keep an eye on, oh, I should use this ontology term in my paper. So because of that, and also on top of that, many ontology terms, people try to develop it to precisely describe a particular biological process, a gene function, or cellular components. So they can be long, they are not natural language, and they are not really appeared in many biomedical literature as the text. So, so it's very difficult to find those terms. And so those are the limitations of this method we developed. However, though, the advantage of this method is the explicit representation, which means you can see that for any gene, for the gene ontology fingerprints, I know exactly what each dimension is. For example, in this vector, the first dimension is lipase activity, the second dimension is carboxyl esterase activity, and the third one is chylomicron. So all these dimensions have an explicit meaning. That's what I meant by explicit distributed representation. And this is important because then you can actually perform some further analysis to find out what dimension is important for your downstream task, like, for example, predict changing relationship, predict pathway membership, all those kind of things. So it has an inherent interpretability. Okay. On the other hand, if you noticed, when I explained to you the difference between one-hot encoding and distributed representation, you can see that in one-hot encoding, each dimension, you still have very clear meaning, right? Each dimension is a word that you know the meaning. However, in distributed representation, you can see that you don't have that anymore. The dimension becomes just numbers. Okay, and that is because when you use neural network or use things like, for example, SVD, you lose this kind of information. So, so after we developed the ontology fingerprints for about 4 years, then people publish papers like on this modern learned deep representation. The most significant one is, I think many people know, it's called Word2Vec. And so in this work, basically, they designed a task to use neural network to learn the distributed representation by, design this task to predict the surrounding words. So in this case, for example, you have this phrase, SMARCB1 deficient bladder cancer. You take deficient, and then you ask the neural network to predict what is before this word, what are the words after this word. And imagine you take, many biomedical literature, you feed that into this neural network, ask the network to, predict their neighbors by, learn the distributed representation. In the end, you will learn this distributed representation that represent this concept very well. Because, obviously, you can see that the context of these words before or after a particular word is actually important and quite really reflecting what this input word is. So that's one very widely used method nowadays. The other approach is called the GLAV model. It's called a global vectors for word representation. So this is a model based on a global log bilinear regression model, and it's co-occurrence based. It combines the advantage of global matrix factorization and the local context window methods, which refers to things like what I just said, right? You predicted the neighboring words for a particular word. So the basic idea is this, right? So you sort of calculate co-occurrence, you calculate the probability and the ratios. So here you have these ice and steam, and then you also have these word like solid, gas, water, and the fashion. And then if you only look at the co-occurrence of these words, right? Like for example, ice, the co-occurrence of this ice with all these different words, you can see the difference is not really, very obvious. And again, for the word like steam, you also can see that, the distribution is quite sort of uniform. It is only when you calculate the ratio of given ice with this particular keyword or given steam with this particular keyword, then you can see a significant difference, right? For example, ice, for ice with solid, it's a quite positive number that's greater than 1, which means, the ice and the solid, these two are quite relevant, right? On the other hand, if you look at the gas and the steam, it is much smaller than 1. And again, that also means these two are quite relevant. So that's the basic idea behind this, to generate this distributed representation using the GLOB model. The third approach is embeddings from a language model. So the obvious example is the transformer model, right? So for us, many of us probably know, transformer is a quite successful and widely used deep learning model for language processing. And also like ChatGPT, GPT-3, those are also the core, also uses transformer. So basic idea is that you have these sentences, and then each word, you have each word in a sentence, and then you have this embedding, and then you feed these input embedding into this transformer model that has what's called a multi-head attention. If you remember, I mentioned that when we compare all against all genes, you generate a table with the similarity. So here, this attention table is very similar. So you have imagine you have a sentence, a stream of words. Each word is like a vector, right, represented by a vector. And then you calculate this attention by compare these embeddings for each word of this input sentence against all the other words, the embedding of all the other words, to generate this self-attention table, and then feed that into neural network. And so multi-head means you do these that will capture different characteristics of your text. And then, you go through several layers of these, and then in the end, you generate the output probability to for your embedding. And so this kind of language model, they can train on large amount of text. They can learn a lot of details in the input from the input data, and the model perform very well on many downstream tasks. Imagine you just think about how ChatGPT perform, and everybody's crazy about it. I mean, good or bad way. But you can imagine that these kind of model definitely capture a lot of important information. Then the question is, can we use the embedding from this language model for certain things we just discussed about? right? So here is an example of what we did. So you take the embeddings from, say, a bird model, like a PubMed bird, right? So PubMed bird is a bird model. People use all the PubMed abstracts trained. So presumably they should learn a lot of information from all these PubMed abstracts, right? And you take the embeddings from that model. Here, I give you an example. Say you have a gene called SMARCB1, the other gene called TP53, and you have this distributed representation. So you can compare these factors to calculate their similarity. And you do that for all the gene pairs. And then you can evaluate to say, this kind of similarity is really biologically relevant, right? So the way you do it is you say pick a particular pathway, and then you take all the genes belong to this pathway, and then you calculate the similarities of gene pairs from this pathway using these learned embedding, and as a control, you randomly pick one gene from this pathway, but pick another gene that does not belong to this pathway. And you also calculate the similarity score, right? So then you have a distribution. You have a similarity score from gene pairs that both genes belong to the same pathway. And you have similarity score for gene pairs that one belong to a pathway, the other does not belong to this pathway. So we know their relationship is not like defined by a particular pathway. You would imagine that if this kind of embedding really reflected the biology, this similarity score should be able to differentiate gene pairs that both genes belong to the same pathway, which we know that these two genes are relevant and perform functions that relate to each other because they belong to the same pathway, versus a gene belongs to this pathway, but the other gene has no information whatsoever, and they don't belong to this pathway. You should see a different distribution that can differentiate these two pairs, but unfortunately, that's not what you see. Instead, here I give you an example that these two distributions, you just cannot differentiate them at all. Okay, so that is a challenge for directly using embedding from this kind of language model. So what we try to do is to take the advantage of this learned embedding in combination with the our idea of this distributed representation from the explicitly distributed representation from our ontology fingerprint work. And here we call it augmented convolutional enrichment representation, ACE. So here is basic idea. So we have symbolic entity encoding. That's basically the fingerprints we sort of created, and then it carries more information, performance, and also it's portable, interpretable, and we use controlled vocabulary. We do the enrichment analysis. And on the other hand, embeddings learned from text from this language model, they're flexible. They perform much better actually in many downstream tasks compared to this fingerprints approach. Obviously because they learn from very large amount of text with very layers of these neurons, but they are not interpretable, right? You can see that I showed you, each dimension, you don't know whether it is relevant to a particular function or, any biological aspects, and it's not very portable. And so these kind of embedding trained with the neural network with labels. So we try to combine these two, and then we sort of generate this new model. So here are the first steps. So basically, we perform the convolutional neural network for enrichment representation. We randomly sample the abstracts with replacement. We do the enrichment analysis, and we adjust the p-value with false discovery rate and then Benjamin Archberg. And then we took the median p-value as the distributional statistics for the representation. And so the second step, then we do the feature extraction, extract the features with deep denoising autoencoder and perform the dimension reduction. In the end, we have this word embedding. And then we perform what we call the feature magnification. We perform the embedding training for the terms using word2vec on PubMed, and then we concatenate these two feature sets, and we perform the feature fusion using autoencoder. In the end, so here's the data source we used, PubMed. We use the genes, we use the disease network, and so in the end, we obtained these distributed representation or embedding, and then now if you calculate the gene pair, the similarity scores, the distribution of the similarity scores for genes belong to the same pathway versus random, you can see a significant separation, which means now this distributed representation is much more biologically relevant. And here are some performance comparison, and you know, our methods have a significant improvement compared to the others. And we apply this approach for COPD, coronavirus relevant disease, and other things. So here's what we identified compared to what people know. One interesting thing here is we identify something called pigmentation that is sort of we think relevant to coronavirus. And then there are some reports. I don't know whether you guys remember. You know, there are pictures of people contracted COVID. They have changed the skin color, those kind of things. We still don't know what it is yet, but it's kind of an interesting observation. And so the other thing I want to just quickly go over is ensemble learning for name entity recognition and relation extraction. So this is what we recently did to participate in this LeetCoin challenge that organized by NCI, by National Institute of Health. That has two parts. One is name entity recognition. The other is the predict the relationship of these entities. And so here are the two steps, and here's the data sets, 400 PubMed abstracts and 100 abstracts for performance evaluation. And here are some relationship. And here are again some more information about the relationship that differentiate novel discovery versus existing knowledge. So not only have you predict this relationship, but you have to tell, is this relationship already published before, or it's a new report? So this is kind of important for many tasks. And so here's our setup: 10-fold, 9-fold for training, 1-fold for evaluating. And here's the highlights of our system design. We use ensemble learning, use the multiple BERT models, and we consider the fold model and the context for prediction. And we also use a technique called cross-sentence input generation. And in the end, our performance for the first task, name entity recognition, reached 0.916, and our team ranked #1. And for the relation extraction, our team ranked #2 as an overall evaluation as well. So So that's my research work here. The last a minute or so, I just want to give you some ideas about what we did for the past 5 years. We established data science informatics core for cancer research. And so this is what we call it, DSeeker. And so basically the idea is that DSeeker are people who perform the act of DSeek, which means, get rid of illness. So the whole idea is basically we have this multi-level biomedical research from population to individual, to organ tissue, to cell molecules, all these levels of data, and for biomedical research. And here are the participating faculties of this core that covers clinical informatics, bioinformatics, biostatistics. And there are 4 major components. We have data, hardware, methodology, and service. And so far, the core is very successful. We helped contribute, publish 127 papers, help people to get like over 58 million extramural grants. And also we build a state-of-the-art computing infrastructure tailored for data science and AI research with lots of GPUs and also large memory server and also a computer cluster. And so with all that, I will stop here and take any questions.
Yeah, thanks everybody for sticking around. I do see some questions, so feel free to leave if you have to go to another meeting. But if you want to stick around, I think we can answer a few questions. So I think I just saw a question in the chat. I'll just read it for your convenience. What do you see as the role for gene fingerprints in data sharing and reuse?
Right. So it's actually quite relevant. We actually thinking about maybe annotating data with distributed representation, right? So, I mean, for data discovery right now, I mean, even though people are doing all these like metadata and controlled vocabulary, ontology, all those kind of things, but in terms of data discovery in computational way, other than say database search, those kind of things, I'm not sure there's a really good way of doing that. So maybe if we have distributed representation for data sets, inferring how these data sets are related, or develop a way to curate this distributed representation to identify the data you want, that might be a good way for data sharing.
I have a couple questions. Thank you very much for this presentation. It's so nice to see that people are working hard to try to make sense of so much data coming from gene studies and trying to relate those to clinical applications. What kind of, if you could elaborate on your concept and term abstraction from those abstracts or papers. So like what kind of text mining tools did you use? Was it strict text with stemming and normalization or NLP? Did you use the mesh indexing, or you looked at more just the text of it that was there?
Right. There are, in terms of that, actually nowadays, there are quite some tools for text mining and NLP and those kind of tasks. And for us, mostly we can develop our own because, as a research group, you have more flexibility, to sort of modify the method, modify the parameters, or incorporate your new ideas into these kind of tools. So we mostly develop our tools on our own. And I mean, certainly not from scratch, right? So there are, like if you look at GitHub and other places, there are, many sort of tools for different purposes. But but you know, so you can use libraries, you can use you know, small tools from.
There's a question from John Osborne. He's our, you know, renowned for his NLP work. So maybe knows more about this space than I do about text mining. What kind of hardware GPU did you use for Litecoin?
Right. So we used so we have this what's called the NVIDIA DGX A100. So right now A100 is the latest GPU from NVIDIA with 80 gig of memory and over 5,000 cores. So the server has like 8 of these GPUs, and it's pretty fast.
I have a question about the, you know, you showed the clustering with the different kinds of functions. You know, was that strict? Was that more clustering, or were you also trying to approximate the location within the cell? Because I kind of got the sense from watching it, that looking at it, that you were incorporating both.
Right. Right now, we only sort of look at everything as a whole to infer gene-gene relationship, right? But you know, certainly because we know what are these ontology terms, we can definitely look into like components versus biological process versus function. But we haven't really looked into that yet. Yeah.
Like you had transport things. I noticed some of those seem to be on the surface, and I don't know if that was just coincidence. Right. Oh, that was just that cluster. In terms of, you mentioned the adjacency. Was that more from the, you know, just the analyzing things, how closely things were together, and both based on gene function or protein function? Did you also consider semantic distance? You know, that like, you know, from, you know, like something's a subtype of something else. Okay, it's probably related.
Right. So I think one thing I mentioned, but I didn't emphasize, right? So like semantic relation or the typical NLP task, you know, basically you take the text, you identify your entity, you identify your relationship or semantic you know, relationship or something like that. These are all explicit relationship that which by that I mean, you know, these relationships are already in the text. The task of NLP or text mining is to extract those relationships, right? So but what we do here with distributed representation is a true inference. What I meant by that is that you take the distributed representation of one gene, you compare that to a distributed representation of another gene, you calculate how similar they are. This process, they infer this relationship. I mean, certainly it's based on the information from the text, but the relationship you inferred is not explicitly expressed or captured in any text. So in that sense, this is a true inference of something that is not explicitly expressed or captured in the text. So that's the difference here. We didn't do any of those semantic relation extraction. Yeah.
Well, you're trying to discover new knowledge. So, right, something's already been stated. Maybe that's not that useful. And you know, it's kind of a model. How about the future? Other ontologies besides gene ontology or different domains?
Right. So we're doing a lot of things, but right now we are very interested in like these embeddings. As you can see, you know, the raw embedding you get from this language model, you know, we also look into large language model. They may be good for a lot of you know, tasks that the model is doing, but just understanding this embedding or, you know, use them to infer relationship, it's not really a good performance. It does not have a really good performance, and we try to understand why. We try to improve these. We also try to understand the embedding as a way to deal with these black box that you know, thing everybody's talking about deep learning, right? So you know, the performance is great, but it's a black box. We don't know all these things. I think if we can develop a way to understand these embeddings, maybe we can have a better or more transparent or white box sort of way to use this deep neural network.
Yeah, I thought it was very profound how you identified all of these proteins that might be involved in a certain process or pathway that had not been previously identified. I think that
Right.
it's all well to just sequence everything you find in the cell, but if you don't know what it does, it's not that useful. So I was wondering, I'm sorry, you were going to say something.
No, go ahead. Yeah.
And you said, oh, look, wow, if we target these things, that's much more effective than, and we didn't even know about that. So, what kind of collaborations are you involved in or planning in which such as people working with drug discovery or drug testing and, getting, farther along on that translational spectrum?
Right. So I think you really you ask a really good question about this, right? So for that sphingolipid work, right? So my collaborator who actually started the entire sphingolipid field, I mean, certainly with a few of his colleagues. So he has been working on this sphingolipid pathway for many years. It's a small pathway, only 30 known genes. When I talked to him about this idea first, he laughed. He said, I have been working on this pathway for many years, and he does not really believe we can find anything new, right? But in the end, we identify these genes and we tested in the lab. Indeed, they can impact the pathway. So I think that's, to me, is very satisfying. And then down the road, I have been collaborating with many people in the lab. So I definitely want to collaborate with people on like drug discovery, drug combination, and as well as some of this evaluation of the drug target. We can identify all those kind of things. Yeah.
Yeah, thanks. Yeah, especially if it was something we already have a drug against, but that nobody ever tried, that knew was related, so they didn't try that combination. Any questions from anybody else? I see there's a few people who have stuck around. You can always contact, afterwards. But I mean, you have any burning thoughts, questions, things right now since he's here?
Yes, I have a
Go ahead.
question. I'm not a computational biologist. I am a physician and scientist. I work on the melanin pigmentation. This is one of the area. And for me, it was very puzzling how this melanin, because you use pigmentation, but I assume it is melanin pigmentation. How this has come and which direction and what is the relationship? I'm expert in this area, so I can help you. But this would be for a longer discussion. But how does melanin pigmentation come out in your equation? It was one of the analyses.
Right. So that's what I mentioned is that, the way we perform this inference is try to see what dimension of this representation is sort of related to the counterpart in another gene or disease. So right now we just identify these, but we haven't really looked into the details of how this is related. So that is something I think maybe experts like you can help to identify. Yeah. So
I think that we may got in touch because you know Jack Chen, so he can contact us, and I can really think and help because there are many dimensions. There is many hormones, peptides we can regulate negatively, positively, and there are also different enzymatic structural proteins involved in this. And melanin pigmentation has a variety of functions and. With the coronavirus, well, it's one which can cross your mind very quickly. If you have increased melanin pigmentation, then there is absorption of UVB, which, for example, induces the production of vitamin D3. And vitamin D3, we know there is a correlation this in the way positive, so it is against the infection. So there are many dimensions, but this could be explored in more trying to do in more educated way.
That's great. Let me take a screenshot of your name. I think I will definitely ask the person who work on this contact you. Yeah.
That's all, it's always amazing how everything's connected, that you think things would not be relevant that are connected. I know there's only a few people left. Maybe, are you at a point where you have a job posting, or you could describe, because you said you were recruiting. So, just
Right.
to
We
get that word out
actually,
there.
yeah, we have, our school, we are, like I said, we are recruiting chairs. We have 3 departments, the data science department, the clinical informatics department, and the bioinformatics department. We are sort of recruiting, department chairs. And also after that, I will imagine each department will recruit faculties as well. So I think I can send you those information, and if you can distribute it, that would be great.
Oh, okay, yeah, yeah. It's like, and you're in Houston, nice weather, and there's a lot of big medical, big hub for medical research, clinical research. Well, okay, do we need your permission to access the website to do search using ontologyfingerprint.org? I get a blank page when I'm doing that now.
Oh, yeah. So I need to check why it shouldn't be a blank page. I need to check that, but the functionality of this right now is kind of limited, because we now move more toward this deep learning based approach. But if you need or have a project that need these kind of things, you can send me an email. We can definitely work with you.
All right. Well, thanks very much. And thank you to thank you everybody for staying late and for the wonderful presentation, and have a great weekend. Hope to see you at the next Power Talk. All right. Thanks again. Bye now.
Yeah. Thank you. Bye.